Okay, so today we're going to talk about DNA. And one of the things that you have to be able to understand about DNA is its shape. And this shape um, is depicted here. This is a very nice, pretty model of DNA. What we have to remember is that there's something called a backbone, which is this area along here. This is called a backbone. So the backbone of the DNA molecule is actually twisted in this shape. It's called a double helix. And a double helix is something that is twisted, sort of like a spring, and it's got these two areas called the backbone. So those double, that's two backbones. And then we have these sections here called the rungs. And we're going to see them in a little bit better detail, but just to give you guys an idea, we need to understand that it's a twisted double helix. This is the backbone, and these are the rungs. And if you remember from our first quarter, we talked about the building blocks of DNA, and those are called nucleic acids. And nucleic acids are made of nucleotide monomers. And if you remember me drawing these nucleotides, remember I said we have a house and a balloon and a flag. That's what you have to remember. And each of these has a specific purpose in the DNA molecule. The phosphate group, which is the balloon, it contains phosphate. And then we have a sugar, which is the little house, and it's a pentose sugar. There's 5 carbons. If you remember, every kink is a carbon. So we've got 5 carbons here. That's a 5 carbon sugar. And it's called deoxyribose in DNA, and it's called ribose in RNA. And then we have what is called a base. And we just talked about the base very briefly in quarter one. Now we're going to actually learn about what the bases are. There are 4 bases, and the bases are guanine, cytosine, adenine, and thymine. And I always tell kids to remember GCAT and little mental model. You guys can have this funky cool cat with his nice sunglasses on. He's a cool cat. He is the G cat. Here we are with our little cat. Okay, and that's how you should remember the bases, G cat, and that will become evident a little bit later if you remember G cat. Okay, now we have to look back at where DNA is located in the cell. We need to remember that we have a chromosome inside of the nucleus, and if we unwind the chromosome from its nice, tidy association with histones, you remember those proteins that help it wind up. If we unwind it, much like we're unwinding our yarn, we unwind it into sections of DNA. And here are those bases that I talked about earlier. There's AT and there's GC. GC, G, C, A, T, GCAT. And that helps you remember the order because G always goes with C and A always goes with T. So that's a good, another good reason for remembering GCAT. Okay, so if we really look at the molecule, we'll see the 5 carbon sugar. There's the house. There's the balloon, the phosphate group, and there's the base, the flag. Okay, and so if we take a look at the way the molecule or the monomers are associated with each other, we see that each of the sugars is attached to the phosphate of the next sugar, and the bases are actually sticking out in the center. And if you look at the other side, the other side of the double helix, remember these are these parts. This is the backbone, and these are the rungs here. And this is untwisted, obviously, to make it easier for you to look at. And if we look, there's all our 5 carbon sugars. There's our phosphate groups. And those bonds between those phosphate groups and the sugars are really nice, strong bonds to keep the molecule in place. And then we have the GCATs. There's G with C and A with T. They actually bond together with hydrogen bonds. And if you remember, hydrogen bonds are those weak bonds. Remember, like a high school romance? And hydrogen bonds are easy to break, okay? And you have to remember that DNA actually is a really nice stable molecule, but we have to break it so that we can replicate it. And we're going to learn about replication in this unit. And here's another look at a little more simple look. So here's our sugar with our phosphates, and here's the backbone all the way up here. It's a really nice strong backbone. And then we have our bases. And if you look, you see G is always with C and A is always with T. That's our GCAT. Here's another look at it, a little bit more simple. And you see that we call this the sugar phosphate backbone. That's another way to remember it, okay? Sugar phosphate backbone. And these rungs are held together with hydrogen bonds. You remember those weak bonds we talked about. This is just a very simplified version of a DNA molecule. So here we are we've got the backbone here, and it's twisted in a double helix. And if you look here, it's actually being untwisted. And when you untwist it, you actually allow it to what we call unzip. So it actually pulls the hydrogen bonds apart. And if you see here, these were hydrogen bonds once before, and they were pulled apart. And when they get pulled apart, we can actually start to replicate it or make a new strand of DNA, which is very important for this unit. So we're talking about making new DNA. Here, once again, we see the sugar phosphate backbone. Here are the rungs with the bases, GCAT. And if you see that we've actually pulled the hydrogen bonds apart and exposed these bases. And this is very important in replication because what we want to do is we want to make a new strand of DNA from our old strand. Here we go again, another picture. So here's the bases that are being added to the old strand. So here's the strand that was pulled apart. And then there's this enzyme called DNA polymerase that comes in and adds bases. Don't forget, poly means many. So we're going to make a polymer with a enzyme. And ASE always depicts an enzyme. So it's an enzyme that makes a polymer. And so DNA polymerase actually adds bases to the old strand, making a new strand. Here's just another picture showing the new bases coming in and being added to the old strand. And here's the old strand that was pulled apart from its other backbone. And here we've got the helix. It's still put together. And this is actually an unzipping enzyme called helicase. And it's like a helix. And what it does is takes it away from being a helix. And here we are, enzyme ACE helicase actually unzips the enzyme. And here's what it looks like in very simplified. There's a DNA molecule with the rungs and the sugar backbone. And the helicase comes in and separates the rungs, splits all of those hydrogen bonds, and then these new bases can come in when the DNA polymerase can come in and add those new bases to the old strand. So we end up with two strands. This part of the strand is from the old DNA, and this part of the strand is new DNA. Similarly, we have new here and old here. And we call this semi-conservative because conservative means to save and semi means half. So we actually save half of the DNA molecule and we make a new one. So it's called a template. This is the template for the new DNA molecule.